This video is about Chapter 3, Cell Division. The learning outcome of this video: at the end of the lesson, students should be able to explain cell cycle, show the stages of cell cycle, and explain the stages in the cell cycle, which include interphase and mitotic phase. According to modern cell theory, it states that all new cells are derived from other cells, which means that all the cells must include the cell division process. According to the Campbell book, 11th edition, cell division means reproduction of the cell, which requires the distribution of identical genetic material, which is the DNA, from the parent cell to the daughter cell, except for meiosis process. So cell division, which means the integral part of the cell cycle, is the life of the cell from the time it is first formed from a dividing parent cell until its own division into two cells. Basically, we have two types of cell division. First is known as mitosis, second is known as meiosis. So the meaning of mitosis is a process of nuclear division in a eukaryotic cell, which include four phases, which is prophase, metaphase, anaphase, and telophase, which results in all the daughter cells having the same number of chromosomes as the parent cell. While meiosis is the type of cell division that occurs in reproductive organ, which include meiosis 1 and meiosis 2, that results in all the daughter cells having only half the number of chromosomes found in the parent cell. Before the cell can divide, we must replicate the DNA first, so that after the process of cell division, both the cells will get the same identical amount and the content of DNA. Then the two copies will be separated. So each daughter cell ends up with a complete set of genome. So now we are going to learn about the process of cell cycle. So the meaning of cell cycle is a complete sequence of events that occurs in the life of an individual diploid cell, which is 2n. Okay, so means haploid cell, they do not have the process of cell cycle. So the cell cycle consists of two main phases. First is known as interphase, also known as non-dividing phase or resting phase. Another phase is known as mitotic phase, which is the dividing phase. So mitotic phase can be further divided into mitosis and cytokinesis, where mitosis is the division of nucleus, cytokinesis is the division of cytoplasm. So cell cycle has two phases, okay, interphase and mitotic phase. Interphase has three subphases, which is G1 phase, also known as first growth phase, S phase, also known as synthesis phase, G2 phase is the second growth phase. While mitotic phase has two subphases, which is mitosis process, which has four substages: prophase, metaphase, anaphase, telophase, and followed by cytokinesis. So this is the cell cycle in the form of diagram, where we can see the interphase. They are almost most of the cell cycle, they have the largest interphase. Okay, the interphase is the longest phase in the cell cycle. Mitotic phase is the short phase in the cell cycle. Okay, so now we look at the interphase process. So interphase is the longest phase in the cell cycle, which accounts for 90% of the cell cycle. So this is the phase before the cell can enter the mitotic phase. Alright, so it is the previous, preceded by the previous cycle of the mitosis and cytokinesis process. So during this interphase, the chromosome is invisible under the light microscope. So the cell grows by synthesis the proteins, organelles such as mitochondria and endoplasmic reticulum. They will replicate the DNA or duplicate the chromosome and prepare for the cell division. So now the detail of the interphase, the G1 phase, S phase, and also G2 phase. Okay, so now we look at the first growth phase or first gap, which is known as the G1 phase. So during this phase, the cell prepare itself. How they prepare themselves? They increase in the size. From a small cell, they grow into a big cell. So how they grow into the big cell? They have to increase the volume of the cytoplasm. Right? They will synthesize the protein, histone protein, in preparation for DNA replication, so that later they have double amount of the DNA. So they need more histone protein. Synthesis of the carbohydrate, lipid, and RNA for the use of other daughter cell, and synthesis of cytoplasmic organelle such as mitochondria and ribosome, lead to the increasing number of organelles. Next is the S phase, which is the synthesis phase. So synthesis phase is the longest subphase in the interphase. Means among the interphase G1, S, and G2, S is the longest phase where replication of the DNA occurs here. So DNA initially they have only one, now they become two. Double amount of DNA. And resulting in the DNA content of the cell being double. So initially DNA is in the form of double helix in one copy. Now they become double copies. So there is double amount of DNA. So chromosome is still in the form of chromatin and become duplicated. Initially chromatin is only one strand. Now they become duplicated. Sorry, I need to erase it. So now they become duplicated. So now they become duplicated. Alright, so the duplicated chromosome, they consist two identical DNA molecules. So before the replication process occurs, the chromosome composed of only single DNA, which is arranged into the fiber, the unduplicated chromosome. After the replication process, each chromatin or the chromosome, they compose of two identical DNA molecule, which is duplicated chromosome. So when the chromosome become more condensed, which is started to condense at G2 phase, they become more visible as two strands that are held together. So at this point, we still can call this as one chromosome, which is composed of two sister chromatids. So this is the diagram that show the double chains of DNA. Then nucleosome, when it is associated with the histone protein. So now in the interphase, okay, before the S phase, our chromatin is only in the single strand. Okay, so after the process of S phase, the chromatin become double. So note that the centromere is in the center here, so they have double chromatin. Okay, because of the DNA replication process, so they have double DNA. So after the duplication of the chromosome, okay, means this is the duplicated chromosome, then you call it sister chromatid, okay, during the mitosis process. So now G2 phase, second growth phase, the cell continue to grow. So note that the word continue here. So just now G1 phase, the cell already grow. So now the cell is continue to grow, energy store increase, so that later when they enter the mitosis, the energy is enough to divide the cell. So they continue to increase in the number of organelles. So again, continue, because this is not the first time the organelle increase in the number. It has already happened in G1 phase before. So now G2 phase, they just continue to increase in the number, such as the mitochondria, Golgi body, and ER, and increase in the size of the nucleus. Okay, so the chromosome starts to condense. Okay, start to condense. So the two centrosomes have formed by replication of a single centrosome. So the cell complete the preparation for the cell division. Chromosome in the form of chromatin, okay, which is duplicated chromosome. So now it is still in the chromatin form. They are not yet become thickened. So chromatin just started to condense. Okay, started to condense to form the visible, shorter and thicker structure called chromosome. So as an overview of the G2 phase, so the nuclear envelope, okay, nuclear envelope bound the nucleus. Each nucleus contains one or more nucleoli. So at this moment, our nuclear envelope and nucleoli is still visible. Two centrosomes have formed by replication of single centrosome. Okay, centrosome is the region which consists of centriole. Chromosomes are duplicated during the S phase, cannot be seen individually because they have not yet condensed. Okay, remember, before the mitosis, our chromosome not yet condensed. So the cell may contain a pair of centrioles in centrosome region for animal cell.